Section 1 of The Psychic Life of Micro-Organisms by Alfred Binet. This is a LibriVox recording. All LibriVox recordings in the public domain. For more information or volunteer, please visit LibriVox.org. Recorded by Leon Harvey. Chapter 1. The Motory Organs and the Organs of Sense. Motility. From the schedule of the groups of animal microorganisms which we have given, it will be seen that they are subdivided into 4 classes: the infusoria, the mastigophores, the sarcodines, and the sporosa. The distinction between these classes depends on the existence and the nature of the motor organs. The infusoria comprise the protozoa and move by the aid of vibratile cilia, distributed in greater or less number over their body. The second class, the mastigophores, comprises those animals which move by the aid of flagella, that is to say, by the help of long filaments. The third class, sarcodines, comprises those animals which move by the aid of pseudopodia, which are projections of the substance of their bodies. The fourth class, sporozoa, is characterized by the mode of multiplication. They are reproduced by spores. In the animals of this group, the special motor organs are wanting. These creatures therefore generally move very little, or they present only movements of which the principles are unknown. We shall successively describe the pseudopodia, the vibratile cilia, and the flagellum. The pseudopod. The formation of pseudopodia takes place chiefly in naked cells, in cells lacking an enveloping membrane, in the sarcodines in general. They can easily be studied in the amoeba princeps, a microscopic animal which is found in abundance in fresh water containing organic matter in a state of putrefaction. It has the aspect of a small gelatinous mass, irregular, formed of a colourless substance, the protoplasm. The chemical nature of protoplasm is still very imperfectly understood. It is only known that it is the result of a mixture of albuminoid matters, with an addition of water and mineral elements. In the protoplasm of the amoeba exists a small rounded and reflecting mass, containing 1 or 2 bright corpuscles on its interior. This small mass is called the nucleus, and the corpuscles the nucleoli. The form of the body of the amoeba is rendered very irregular by the fact that certain parts of the mass lengthen and form short and rounded protuberances, which are designated by the name of pseudopodia. It is by means of these pseudopodia that the animal moves. It emits them in the direction in which it is going, then it retracts them, while other parts of the mass are in their turn elongated. The whole body moves by creeping. The amoeba in moving has the aspect of a drop of oil moving along. To explain the mechanism of this movement, it must be supposed that the extended pseudopod seizes some point of support with its free end, then in contracting draws the entire mass of the body up to this. But it is difficult to understand what the cause of the elongation of the pseudopodia is. It has been supposed that the protoplasm is endowed with great elasticity, and that the elongation is the return of this substance to its primitive form. That is not the explanation given by M. Roget. The learned professor of the museum has been kind enough to write out the following note for us, in which he recapitulates his opinion. Every time that a protoplasmic organism dies, or is subjected either to a strong electric excitation or to a relatively high temperature, plus 45 degrees Celsius to plus 50 degrees Celsius, the pseudopodia are retracted and re-enter into the mass, which assumes a globular form. The same is the case in the protoplasm of vegetable cells, the intercellular reticulum of which breaks in receding, or else the mass of protoplasm divides into spherical bodies. These states of retraction are the analogues of muscular rigidity, and, like it, represent the condition of maximum contraction in the protoplasm. Nevertheless, the style of the vorticels, Cartesium, which is a protoplasmic formation under the same conditions, remains in a state of permanent retraction. It follows from this that the emission of the pseudopodia, their elongation, cannot in any case be considered as a direct act of the contractility of the protoplasm. The production of the pseudopodia, one of the most difficult problems, cannot in my opinion be explained except in the following manner. All protoplasmic masses, and especially the amoeba, consist of 2 parts, an enveloping membrane or ectosarc, viscous and elastic, and the central liquid contents holding the granules in suspension. From the time of the apparition of pseudopod, a current of liquid is visible which penetrates into the pseudopod, and which seems to contribute to its elongation. It is very evident that the liquid is passive, that it penetrates into the pseudopod only because pressed upon from all sides it finds less resistance there. I think that in the appearance, homogeneous hyaline substance of the pseudopod is also a species of hernia of the estisarch, resulting from a diminution of the elastic resistance at the point where it appears, with an increase of elasticity or of contractility, to me two motorolites of the same property, in those parts of the ectosarch where pseudopodia are not produced. When the contractility or the elastic tension of these parts diminishes and returns to its original state, the pseudopod re-enters into the mass. Add to this that, in an amoeba of large dimensions, Amoeba terricola, it has seemed to me that the most external membrane of the ectosarch showed striae of a granular appearance, which may be identical with the striae or contractile fibrils of the ectosarch of the ciliated infusoria, Stentor, Spirostomes, Brusaria, etc. May 20, 1887. The pseudopod does not represent a permanent, differentiated organ of locomotion. It is produced by a simple prolongation of the mass of the body, which can take place at any point whatever, and when the act of locomotion has been accomplished, this prolongation re-enters into the common mass without leaving any trace of its emission. In other animal species, for example, the Petylobus of Lachmann, initial traces of differentiation of the pseudopodia have been observed. They always form at the same point of the body, on a level with the anterior part. But in spite of this constant localization, the motor organ has only a transitory existence. It is produced at the moment it is needed, disappears into the mass of the body when the movement has been executed. In the actinopheres there is still greater progress. The numerous pseudopodia emitted by this animal, and which have the form of filaments, are permanent organs with definite functions. The vibratile cilia. The vibratile cilia are short, extremely thin, homogeneous filaments, which are agitated by a vibratory movement. These are distinctly differentiated organs of locomotion. They have, moreover, several functions. Firstly, they enable the animal to move about in the liquid. Secondly, they serve it as an organ of prehension. Thirdly, they permit a renewal of the water which furnishes the necessary air for respiration to the animal. Perhaps they also serve as organs of touch. The vibratile cilia lend to the infusoria their peculiar character, and enable them to be distinguished from all the other protozoa. Cilia are also found in vegetable species when young, in the larvae of colinterets, of mollusks, and of worms. But among the protozoa, it is the infusoria alone that are ciliated. The cilia are distributed in various manners, differing according to the species. In the holotricha, they are distributed regularly over the whole surface of the body, and almost all of the same length. In the heterotricha, they also cover the whole surface of the body, but they are unequal in length. To this group belong the stentors, which have long cilia inserted around a circular surface, extending almost to the mouth. This surface is a rotatory organ, analogous to that of the rotifers. It produces eddies in the water, and thus causes the flow of foreign bodies to the mouth. These animals have the rest of their bodies covered with fine cilia. In the hypotrichia, the cilia are located on the ventral surface of the body and aid in locomotion. In the peritrichia, they form a circular or spiral row on the anterior part of the body, and lead to the mouth. This is observed in the vorticals, sessile species which have no other cilia than those which are used for the prehension of food. The rest of the body is bare. Much has been said about the morphological significance of the vibratile cilia. Several micrographists have held that the cilia are attached to the enveloping membrane only, and have no connection whatever with the protoplasm. That was notably the opinion of Robin. It is entirely wrong. The cilia are never simple prolongations of the cuticle. They have their root in the protoplasmic substance. They pass through orifices in the cuticle, which consequently is pierced by a multitude of small holes. Engelmann, in recent observation, has been able to trace the extremity of the vibratile cilia into the interior of the protoplasm. He made this observation on the marginal cilia of the Stylonychia. From each of these threads he has seen separate a pale fibre, which moves along almost directly beneath the cuticle in a direction perpendicular to the lateral edge of the body. Towards the median line of the ventral face, the fibre is often laid bare, because the body of this infusory voids its protoplasmic substance. There the fibres have the aspect of tight threads. Engelmann sees in this observation a confirmation of the opinion that the bodies of infusoria are formed of a single cell, because according to other observers, there exist also in vibratile cellules filiform striae, which seem to be a continuation of the cilia, and which traverse the protoplasm of the cell throughout its whole length. We might add to this direct observation several other facts showing that the vibratile cilia are indeed prolongations of the plasm. Under the action of reagents, the cilia act like the ciliar protoplasm. They are coagulated by the acids and dissolved by weak alkalines, while the cuticle offers a greater resistance to these same agents. These vibratile appendices are not without analogy to the pseudopodia of naked cells. Dujardin, a French naturalist, demonstrated this in 1835, although efforts have since been made to bestow the honour of this discovery upon the Germans. Dujardin has proved that the amoeboid movement and the ciliary movement are only two manifestations of the contractile power of protoplasm. In fact, if instead of examining the pseudopod with lobed outline like that of the amoeba, we observe the slender filamentous pseudopodia of the foraminifera, we see that the extremity of the filament is agitated by the same vibratory movement as the vibratile cilium. All the transitions from the fine and delicate cilia to the large cilia, tapering in form like a stiletto, which have been called cirri, have been observed. Moreover, these cirri are formed of agglutinated cilia. By the aid of certain reagents they have been dissociated. An observation of the ciliatory infusory, the Didinium nascutum, see the illustration further on, made by M. Balbiani, shows that the movement of the cirri is not an involuntary movement, like that of the cilia or the vibratile filium, with which has often been compared, but that is completely under the control of the will of the animal, like the organs of locomotion of animals much higher in point of organization. The Didinium has two rows of equal and rather strong vibratile cilia, disposed transversely around the body in the form of two belts or crowns. The rest of the body of this animal is entirely stripped of cilia, but its double vibratory belt suffices to enable it to execute the most rapid and most varied evolutions in the water. Not only does it swim forwards and backwards with perfect ease, but the progression in both directions is always accompanied by a rapid rotatory movement of the animal about its longitudinal axis, similar to that observed in other infusoria that have a cylindrical body. The two rows of cilia always act in union during their locomotion, and the direction which the animal gives to them determines the direction in which it wishes to move. In the movement forwards, the cilia are directed toward the anterior part of the body, Figure 1. When it swims backwards, they are reversed, Figure 2. The infusory thus rapidly makes its way across the field of vision by jerks. From time to time it suddenly stops, all the time continuing to turn around rapidly on its axis on the one spot, during which the movement the ciliated belts beat the water in opposite directions, the anterior ones being turned forwards and the posterior turned backwards, Figure 3. The result of this is that the effects of these small locomotive apparatuses neutralize each other in the same manner as two helices acting in opposite directions, and that the animal remains stationary, while all the time turning rapidly about itself, sometimes horizontally, sometimes vertically on its conical appendage, just as on a pivot. Certain infusoria, for example, the Condylostoma patiens, which has been thoroughly studied by M. Maupas, possesses at the same time the two kinds of appendages, the cilia and the cirri. The former, which cover the dorsal surface of the animal, are fine, very dense, and animated by a rapid and unceasing vibratile movement. The cirri, which cover the ventral surface, are placed apart. Furthermore, they do not vibrate rapidly. Their movements are slow, and when the infusory moves, one can see them move successively on the plate of glass, and support themselves there, in the manner of a foot, to make the body advance. When the animal stands still, the cirri are absolutely immobile, while the cilia continue their vibratile movement. This observation, which can equally well be made of the oxytrachid, shows that the vibratile cilia are the organs of involuntary movement, and that the styria are more directly subject to the will. The fact is demonstrated by the experiments of Rosbach, who observed that under the influence of the falling of the temperature from +15 to +4, or of the rising of the temperature from +35 to +40, or under the influence of various chemical substances, the large cilia, the organs of voluntary movement, are paralyzed while the fine and delicate cilia continue their movements, which do not seem to be under the influence of the will. These movements alone cause the whole body to rotate until the vibratile cilia are in their turn paralyzed. Besides the cilia and the cirri, other appendages in the form of membranes are found among the infusoria, appendages which are attached to the anterior part of the body or the peristome. These membranes serve the purpose of causing eddies in the water which bring the floating alimentary particles into the mouth. They are modifications of the vibratile cilia. These membranes, like the cirri, are formed of agglutinated cilia. The flagellum. The study of the third organ of locomotion, the flagellum, brings us to speak of the class of mastigophores, and more particularly of the group Flagellata. The flagellates are protozoa of very small size, all in all very much smaller than the ciliated infusoria. They have no vibratile cilia at all, but they are always equipped with one or more filamentous appendages, which have the form of a long lash. This is the flagellum. This lash, like all the organs of locomotion hitherto studied, has two functions. It is at once an organ of locomotion and an organ of prehension. The flagellum is most frequently single or double, see Figure 4 representing the Euglamendeses with its single flagellum. Sometimes a person can count a much larger number of them, 4, 6, 8, 10, and more. As regards the insertion, the same variations are met with. Sometimes the flagella are very numerous and seem to be planted on the same point of the surface of the body, thus forming a brush or plume. In other species we find several flagella arising from the anterior extremity of the body, directed towards and also posterior or caudal filaments, which are turned towards the rear. This is observed in the genus Trichomonas. The anterior flagella serve for purposes of locomotion, perhaps also for the prehension of food. The posterior flagella, on the contrary, are solely organs of locomotion. They resemble a trailing tail and perform the functions of a rudder. In passing, we may point out the great morphological resemblance between the flagellata and the spermatoza of animals, the antherosa and the zoophores of plants. The organs of propulsion in these beings are the same. The protozoan with its flagellum executes the most varied movements, moving first in one direction, then in another, and in different planes. Sometimes the animal curves about entirely, but most frequently, when he uses it as an organ of prehension, he extends its whole length before himself. The basilar part remains completely immovable and rigid, while the free end alone executes movements destined to drive food to the mouth, which is generally situated at the base of the flagellum. Ehrenberg gives to the flagellum the name proboscis. Its peculiar mobility renders it worthy of this name. The flagellum, like the vibratile cilium, is an expansion of the protoplasm through the enveloping membrane. M. Sertz has observed a protozoan, the flagellum of which between whiles re-entered into the mass of the body, and which is mingled. It was replaced by a pseudopod, which soon attenuated and took the form of a flagellum. Bucci has recently made a very interesting observation on this organ of locomotion. Under certain circumstances, the Peridinia, dinoflagellates, throw off their long flagellum and enter into a state of repose. They generate them quite as easily. In the Glenodinium cintium, Bucci has seen the flagellum roll itself up first like a corkscrew, and suddenly detach itself from the animal. Having become free, it stirs about in the water for several minutes before becoming motionless. This observation enables us to refute those naturalists who believe that the vibratile cilium is an appendage of the cuticle, by bringing forward the fact that when the cilia with the portion of the cuticle in which they are inserted are separated from the cell, the cilia continue to move. We have just seen that the flagellum moves even after it is separated from the cuticle. This persistence of movement is sufficiently explained by the protoplasmic nature of the cilia and of the flagellum. From another point of view, the observation of Busschi gives us a curious example of the phenomena of autonomy, which have recently been studied by Frederick. The pseudopodia, the vibratile cilia, and the flagellum constitute the three motor organs that are most frequently found in the kingdom of the protista. Among the infusoria, moreover, particular differentiations of the protoplasm have been described, which may be compared to the muscular fibers of the higher animals. The vorticillae are supported by contractile peduncles. These are filaments capable of rolling themselves up into a form of corkscrew when the animal is disturbed. Certain infusoria can modify the form of their body by sudden contraction. They have been called metabolic, such as the stentors, the prorodons, the spirostomes. In contradistinction, those which do not change their form, for example the paramecia, have been called ametabolic. According to the observations of Liebrekunde, which date back to 1857, the metabolic infusoria have their bodies divided into large granulous bands, separated by bright filaments. It has been asked which is the contractile element. Is it the band or is it the filament? Oskar Schmidt, Kolliker, Stein, and Ruget think that it is the band which is a contractile element. This opinion is based on the following fact, which M. Ruget was the first to observe. At the moment at which the animal contracts, the band presents transverse striae. This appearance is due to the fact that the bands contain in the state of rest small granules which, during the contraction of the animal, are disposed in transverse series, so as to recall the sacrous elements of Bowman. Liebrekunde, Grief, and Engelmann attribute the active part to the bright fibre. Engelmann has based his opinion on the fact that he recognized in the filament the property of double refraction, which according to him belongs to all contractile substances, while the substance which separates the filaments shows only single refraction. However that may be, it is one of these two elements that possesses the power of contraction, and which deserves the name of myophane, which Haeckel gave it. It is very remarkable that in the stentors and the spirostomes, the fibrillstratae are in intimate connection with the basilar extremity of the vibratile cilia. In the vorticillae, one can clearly see the fibrils converge towards the axis of the style, the contractile element of which they constitute. We shall not leave the study of the motor organs without saying a word about the rhythmical movements which can be seen in the contractile vesicle of the microorganisms, vegetable as well as animal. This vesicle is a small cavity which is dug into the protoplasm, and which alternately increases and diminishes its capacity. Scientists by no means agree as to its exact function. Busschi and Stein consider it to be a secretive apparatus. Its pulsations are very regular. Their number is constant in every species. In the Chiloton coeculis, a pulsation occurs every 2 seconds. In the Critochium nigricans, every 3 seconds. In the Vorticillae, every 8 seconds. In the Eupots, every 28 seconds. In the Acinerae incurvata, every 6 minutes. Rosbach, whose curious experiments with the vibratile cilia and cirri we have already cited, has made analogous experiments with the contractile vesicles. He observed especially that under the action of alkaloids, the contractile vesicle ceased pulsating in diastole, and dilated it enormously. But poisonous agents do not act all at once on the movements of the vesicle. They begin by paralyzing the larger cilia, which are under the influence of the will. The movements of the vesicle, like those of the small cilia, persist for a much longer time. M. E. Morpus has seen paramecia, killed by a discharge of trichocysts, become completely immobile with their vibratile cilia inert and rigid, while the contractile vesicle continued to pulsate with the same activity. This activity continued for an hour. We have now briefly examined the morphology of the motor organs or motor organisms. it is very difficult to determine the physiological process of the movements produced by these organs. The simplest movements are the ones most easily understood, are those by which a cell suddenly and strongly irritated withdraws its prolongations and assumes a spherical form. This change of form can be explained by a quick condensation of the protoplasm, which becomes the seat of a phenomenon similar to that of a contracting muscle. The sudden modifications which are observed to take place in the form of the so-called metabolic infusoria are in this way explained by analogous phenomenon, so much the more evident as the infusoria which possesses this property show in the cortical layer of their protoplasm ectosarc granular bands which have with more or less justice been compared to the muscles of the higher animals. The displacements of the body determined by the pseudopodia, by the vibratile cilia, and by the flagellum are much more difficult to interpret. Meanwhile, it is probable that the movement proceeds from the contractions of the protoplasm which are produced either in the ectosarc or in the motor organ itself. The latter is automobile, as is seen, for example, when a flagellum separated from the rest of the body continues to move in the liquid. It is well known that any number of discussions have been raised as to the manner in which the pedicle, on which the vorticellae are mounted, contracts. Still more obscure is the oscillatory movement of the bacteria. These small beings are very mobile when they find themselves in a liquid. They frequently exhibit a movement of oscillation which sometimes carries them forward, sometimes backwards. An attempt has been made to explain these movements by postulating the presence of organs of locomotion, extremely slender filaments planted at one of the extremities of the bacteria like small rods, but the existence of these organs has not been absolutely proved. Even more obscure is the movement observed in certain gregarines. It would seem that in the case of these animals, which are often of considerable size, one ought to be able to understand the principle of their movements much more easily than in the case of such small beings as the bacteria. But this is not the case. The polycystids have a very peculiar manner of moving. The motion is one of perfect translation, uniform and rectilinear. The animal seems to slide all of a piece over the object plate. It can go to the right, to the left, stay its motion and resume it again. It is free in directing its movements. Now, during this movement, nothing can be seen to take place in the body from within or without. An analogous phenomenon is to be observed in the diatoms. Some scientists have wished to explain the mysterious motion by translation executed by the gregarines as being due to an imperceptible undulation of the sarcode. But if there were any undulations whatever, one ought to observe a correlative movement in the granules inside. Now, this is something that is never seen. Thus, there still exists a great deal of obscurity concerning the principles determining motion among the proto-organisms. The theories based upon muscular contraction that have been propounded from observing higher animals are by no means sufficient to explain the phenomena of motility among certain protozoa and protophytes. Nervous system. Hitherto not the minutest trace of a central nervous system has been found in a single proto-organism. The nervous function among these inferior beings devolves upon the protoplasm, which is irritable, which feels and which moves, and which, in certain species, as we shall see later on, is even capable of performing certain psychic acts, the complexity of which seems quite out of proportion to the small quantity of ponderable matter which serves as a substratum to these phenomena. There is, moreover, no occasion to be surprised that an indifferentiating mass of protoplasm should be able to exercise the functions of a veritable nervous system. In fact, every nervous element is nothing else than the product of protoplasmic differentiation. The protoplasm embodies in itself all the functions that, in consequence of an ulterior division of labour among the pericellular organisms, have been assigned to distinct elements. It has rightly been held, therefore, that if no nervous system anatomically differentiated existed in proto-organisms, it must be admitted that their protoplasm contains a diffuse nervous system. Among all the observations that uphold this idea, we must cite one to which M. Gruber, a professor at Freiburg in Breisgau, has recently called attention. This observation was made on a large ciliated infusory, the stentor, of which mention will be made so often hereafter that it will be advantageous to give a full description of it beforehand. The stentor has an elongated body, broadened in front like a funnel, and able to fasten itself by its posterior extremity. The edge of its peristome is covered by a belt of vibratile cilia disposed about a spiral line. The mouth occupies the most sunken part of the peristome. The body of the animal is striated with longitudinal bands. At the plane of the peristome, these bands take a different direction. They become transversal and spiral. In the interior of the protoplasm can be observed a contractile vacuole and a nucleus like a string of beads made up of a large number of grains. This infusory, like all the ciliates, multiplies by fission. A contraction is seen to take place in the middle of the body. The segment below the contraction generates a peristome similar to that of the upper segment. Then a second contractile vacuole is formed, and soon the two segments represent two complete animals, which possesses all their organs. Nevertheless, the two stentors continue to be united for a certain length of time by a bridge of matter, located even with the point where the contraction took place. This bridge of matter gradually grows thinner and thinner and becomes as fine as a thread. See Figure 5. Now Gruber has observed that the two stentors united by this bridge or protoplasm exhibit perfect harmony in their movements. They always sway in the same direction at the same time. And this harmony is necessary, because the least contrariety of motion would suffice to break the feeble bond that unites them. Moreover, their vibratile cilia beat in unison. To explain this concordance in the movements of the two animals, Gruber assumes that the entire mass of their protoplasm performs the function of a diffused nervous system, which has the effect of regulating their movements and of making them harmonize. We might add that the infusoria possesses not only a diffused nervous system, but that they must, of necessity, possess special nerve centers endowed with different functions. It will be remembered, in fact, that under the influence of certain poisonous agents, death is not simultaneous throughout all parts of the organism. What ceases first are the voluntary movements of the large cilia. The movements of the small cilia are able to persist much longer. And finally, when all the cilia have become immobile and rigid, the vesicle has still been seen to pulsate for an hour. This gradual death recalls what we remark among the vertebrates. Under the influence of poisonous agents, the brain dies first, then follows the marrow, and lastly the bulb, which is the ultimum moriens. The organs of sense. All the microorganisms are endowed with sensibility. Some, like the infusoria, have exceedingly sensitive powers. But hitherto, organs of sense anatomically differentiated have been found in only a very small number of species. Generally, the protoplasmic expansions, which we have above described under the name of pseudopodia, are regarded as fulfilling the function of rudimentary organs of touch, which advise the microorganism of the presence of objects which happen in its path. But these pseudopodia, which at the same time serve as motor apparatus, do not exhibit any structure which especially fits them for the reception of sensory impressions. Similarly, Stein considers the vibratile cilia as organs of touch. All these are organs which have not undergone any differentiation. We should not stop to consider them. The infusoria belonging to the genus Cryptochilium morpus carry at the posterior extremity a long rigid bristle, which M. morpus regards as an organ of touch, intended to advise the animal of the approach of other infusoria. We shall speak more at length of the organ of sight. This has been the subject of numerous treatises, some of which are quite recent and of the greatest interest to general physiology and psychology. Of all the organs of sense, the eye is the one which is first differentiated. It is found in the organisms belonging to the vegetable kingdom, as well as in those belonging to the animal kingdom. While these small beings do not seem to possess any organ especially adapted by its structure for the reception of tactile, olfactory, or gustatory impressions, a large number already exhibit an ocular spot. That is to say, a differentiated organ for the purpose of sight and for no other purpose. Let us first turn our attention to the eye of the protozoa. It is chiefly in the group of flagellates, and principally in the species that are coloured green by chlorophyll, for example, the euglinae, that ocular spots are found. These spots, which are coloured a bright red, present themselves very clearly to the observation, for they are set off by the uncoloured plasma of the anterior part of the body where they are generally located. Oculiform spots are also found in the species coloured by yellow chlorophyll, Uroglena volvox, etc. Generally there is only one spot, situated at the base of the flagellum. This is seen especially in the Ilona Gheorghiesh, a small flagellate in fissory, which is very abundant in fresh waters, which it often covers with a thick green coating. In the Cyanura, a colony-forming flagellate, there exists in each individual in the anterior part of the body numerous spots varying from 2 to 10. Below we give an illustration representing the anterior extremity of the Ilona Gheorghiesh. According to Klebs, a large ocular spot is noticeable in continuity with the contractile reservoir. Ehrenberg, deceived by the appearance of these two organs, had taken the contractile reservoir for a nerve ganglion. It is not only in the large group of protozoans that the red spots are met with. They are found also among the vegetable microorganisms. A large number of green zoospores exhibit at the anterior, and usually colourless, extremity of their bodies a small red point which seems to have exactly the same structure as the red spot of the Euglena. It was on this fact that Stein based his opinion that the spot of Euglena is not an eye. To him it seemed impossible to admit that the vegetable proto-organisms could possess a vital organ. This is an excellent instance of a priori reasoning. Later on we shall see that Stein's view has now been completely abandoned. The very opposite view is taken, for the eye of the protista is considered as being destined to perform chiefly a vegetable function. Klebs was able to study the structure of the ocular spots by employing a very ingenious artifice. When the Euglena are treated with a solution of sea salt, in the proportion of 1 part to 100, an enormous dilation of the contractile vesicle which forms a hollow in the protoplasm of the animal is induced. Now as the red spot is, so to speak, glued to the vesicle, it undergoes the same dilation as the latter does, thus greatly facilitating observation. By this treatment it has been observed that the spot is a small discoid or triangular mass of jagged and irregular outline. It is formed by 2 material parts. For the base it has a small mass of reticular protoplasm, and the meshes of the protoplasm there are small drops of an oily substance coloured red. This red pigment, which has received the name of hematochrome, is not without its analogy with the green pigment of the chlorophyll, because this latter becomes red under certain conditions. For example, the chlorophyll pigment which fills the entire body of Hematococcus pluvialis becomes red when the animal enters it into a state of rest. The stagnant spores of the algae also assume a red tint. So also in numerous plants, the parts of the flower destined to become red are green as long as they are enclosed in the bud. It is thus probable that the red pigment of the euglenoids is derived from a green pigment. What is the physiological significance of these spots? Ehrenberg considered them as eyes, hence the name Euglena, word for word, pretty eye, which he had given to the species of flagellates provided with ocular spots. This interpretation has been questioned by all the authors of his time, especially by Dujardin. At the present day, however, naturalists have come back to it, in consequence of observations which have been made on other microorganisms that possess a more perfectly developed eye. M. Pouchet has discovered in the Glendonium polyphemus, which belongs to the group of Peridinia, or dinoflagellates, according to the classification of Butchley, an eye about the function of which there can be no mistake. This eye occupies a fixed place in the cellule of the peridium. It has a uniform location and position. It consists of 2 parts, the one a veritable crystalline humour, the other a veritable choroid. The crystalline is a strongly refracting hyaline club-shaped body, rounded at its free end, which is always directed forwards, while the other end is immersed in the mass of pigment which represents the choroid. This latter is clearly determined. It forms a sort of hemispherical cap, enveloping the posterior extremity of the crystalline. In one of the 2 forms of Glendonium polyphemus, the choroid pigment is red, in the other it is black. M. Pouchet has been able to establish that in the young animas the crystalline is first formed of 6 to 8 refracting lobes, which are merged into each other in order finally to constitute one unified mass. Also the choroid is the result of a combination of the pigmentary granules, which at first sparse, group together, and finally form the hemispheric cap that covers the posterior extremity of the crystalline. In fact, the visual organ of the peridium is composed of exactly the same parts as the eye of a metazoon, with one exception, the absence of the nerve element. This is not at all differentiated, but remains diffused like the whole nervous system. M. Bouchet calls attention to the interest which his observation affords from a taxonomic point of view. The Peridinia have sometimes been classed among the vegetables. The presence of starch and of cellulose in the protoplasm has induced Warming to classify them among the Diatomaceae and Desmiticae. He has admitted today that certain Peridinia possess an eye, an organ which has hitherto been considered as the exclusive attribute of animals. Nothing more clearly emphasizes the altogether artificial character of the distinction between animals and vegetables than the results of dealing with microorganisms. Before leaving the Peridinia, we would remark that these small beings afford an interesting fact from the point of view of the history of the protozoa. They are provided with a long flagellum. They exhibit, in addition, an equatorial line on which formerly a crown of vibratile cilia was thought to be recognizable. This supposed coexistence of a flagellum and of cilia had determined the naturalists to form a group of cilioflagellates, serving as a transition between the flagellates, properly so called, and the ciliates. Since then it has been discovered that the Peridinia do not possess vibratile cilia. What had given rise to this error is the presence of a second flagellum on the level of the transverse line, which we have just described. The movements of this flagellum have the appearance of vibratile cilia in motion. Sometime before the investigations of M. Bouchet, M. Kunstler of Bordeaux had discovered in the flagellate of the genus Phasis a red eye which is also formed of two parts. It is composed of a homogeneous globule acting as a crystalline humour and surrounded by a red pigment acting the part of the choroid. Before M. Kunstler, Fleparade and Lachmann, in their important work on Infusoria and Rhizopods, had described a similar visual organ in the Frida elegans, a ciliated infusory of the family of stentorines. Immediately behind the point of trunction, say they, there is found a lunate spot of intense black, evidently belonging to the category of those phenomena which M. Ehrenberg, in the Ophryculae, for example, calls an eye for an ocular spot. The significance of this spot has never been known. It was often very much denser than that of the Ophryculae, and sometimes they would discover behind it a very transparent corpuscle, which involuntarily gave rise to the mind to the idea of a crystalline humour. We cannot, however, add much of importance to this idea, since the functions of a refracting apparatus must necessarily remain problematic, as long as we do not discover behind it a nervous apparatus fitted to perceive the impressions received. This last conclusion seems to us excessively cautious. The coexistence of a pigment and of a crystalline humour aptly suffices to characterize a visual organ. As to the nervous apparatus susceptible of perceiving impressions, it is replaced by the protoplasm, which, is well known, is sensitive to light. Even before that, in 1856, Lebrikun had discovered in a ciliated infusory the Panophrys flavicans, an ocular spot composed of a convex crystalline humour having the form of a watch crystal enveloped by pigment and placed on the convex side of the oral fossa. In another species, the Ophryoglena atra, he found black pigment but no crystalline humour. It is impossible to believe that these organs are not eyes, for they have the same structure as the eyes of comparatively higher classes of animals, such as certain worms, Turbellaria, rotiferous, lower-class crustaceans, etc. All these organs are similarly formed of a small crystalline globule enclosed in a small mass of pigmentary matter. The identity of structure naturally leads to the assumption of the identity of functions. The eye of the euglena is the simplest of all. It is even reduced to the maximum point of simplicity, as it is composed of a spot of pigment. What induces us to believe that this spot is a visual organ is the presence of this pigment. In fact, this pigment is found in the most elementary visual organs. A second argument might be advanced. The red pigment of the euglena exhibits the same reactions as the colouring matter that fills the rods of the retina in the vertebrates. From among these reactions, common to both, we cite the decoloration under the influence of light. Capranacca. Whatever the case may be, one thing is certain, namely that the euglena is very sensitive to the light. When they are kept in a vessel, they are invariably seen to cover the side exposed to the light. M. Engelmann has observed that light acts very strongly upon the small animal. It does not act directly on the spot of pigment, nor, as was formerly thought, on the flagellum, but on the protoplasm which is located in front of the spot. The special microspectral object class that M. Engelmann constructed enables us to see that the euglena always congregate in the band F to G of the spectrum. So far as the vegetable microorganisms are concerned, we have already mentioned that a large number of the algae zoospores exhibit, in the interior part of the body, ocular spots of a beautifully ruby color. These are organs that probably have the same structure as the red spots of the euglena. Moreover, it is probable that certain microphytes possess more complex visual organs composed of red pigment and of a crystalline humor. M. Balbiani has recently testified to this fact. In the case of the Pandorina morum, a spherical colony of green microorganisms, in each colony there exists a certain number of individuals which possess a red spot, the shape of which is perfectly circular. If this spot be examined under a glass of very high magnifying power, one can readily see that it is formed of a small spherical lobule, covered, on a portion of its surface, by a cap of red matter. This observation is all the more interesting because it is made on a being, the vegetable nature of which today is no longer doubted. The Pandorina are vulvacinae which modern botanists place among the algae. We are glad to give our readers the earliest communication concerning this fact. In describing the eye of the protista, we said that the eye is the only organ of sense which is directly differentiated in these lower beings. But perhaps this assertion is too sweeping. Some species appear armed with small organs which could easily be invested with a sensory function. In this respect we may cite the Loxod's rostrum, a beautifully ciliated infusory remarkable for its proboscis and for the muscular sheath which closes its mouth. This animal exhibits along the dorsal surface a row of small organs which by their structure seem destined to act a part in performing the functions of hearing. They are formed of a vesicle, the centre of which is occupied by a refracting globule. They are called the vesicles of Müller, after Johannes Müller, who discovered them. The auditory organs which have been observed in worms and the Coelenterata are apparently composed of a vesiculiform capsule enclosing a solid concretion, called otolith. Thus it is possible that the vesicle of Müller may be auditory vesicles. Up to the present time this organ has not been met with any other species of protozoa. End of section 1.